Hi, I'm here with Carsten Voss, who is the Applications Manager for Europe. Carsten, how is the global biopharmaceutical market developing in respects to medical indications and therapeutic target molecules?
So when you look at the biopharmaceutical market, you have to distinguish between the different regions in the world that you're looking at. So in developed countries like Europe and the North Americas, you are looking at developments for target proteins that are approaching very investigational new drugs, while in other regions like the Asia-Pacific regions, Middle East and Africa, or Eastern Europe, especially in Russia, you have a very strong market for biosimilars. Of course, mainly looking at drugs that are addressing the typical diseases like cancer, diabetes, indications like Alzheimer's diseases and related ones. And of course you have all the varieties of infectious diseases. So in the latter part, a lot of new challenges come up there, but the main part to distinguish is really between the biosimilars and the investigational new drugs. Europe, for example, also has a focus on the biosimilars as well, but this is kind of to differentiate between these regions and also regions like Eastern Europe or the Asia-Pacific regions, for example, like China's. The latter ones, countries develop biosimilar processes to serve their own markets there. So they supply their own needs there. They won't afford the cost-effective drugs, even the cost-effective biosimilars that are produced in Europe and North America. This is the reason why they want to have their own economic processes to supply their markets there. With respect to investigational new drugs, of course, you have a lot of new developments with respect to the target protein structure. A lot of the market proteins is of course dominated by monoclonal antibodies, but the landscape's also slowly changing. So a lot of target molecules now are still having like the same focus there. They're antibody derived. You will find antibody fragments, single chain FV fragments that are coming out of IgGs, and these of course have different requirements for purification.
So what challenges for biopurification results from these challenges?
If you look at new drugs that are developed, like antibody fragments or different antibody fragments evolving from IgGs, non-IgG antigen binders, you have to look at completely new purification schemes. So these are molecules that are of course antibody derived, but they are not handled by the typical purification schemes that is done by protein A and subsequent chromatographic steps there. In this case, you have a non-protein A purification process, so you can't rely on a platform technology and have to develop all over again. For other markets on the biosimilar, you are taking more perspective on the economic benefits you're having. So processes that were developed for the original pharmaceuticals can now be developed with new technologies. They can be redeveloped, they can be made economically more feasible for the companies there, and in this way you are capable of developing a cost-effective drug for this purpose there when they go off patent finally. And that's also an opportunity, of course, for developing countries. When you look at China, Russia, or Middle East and Africa, these countries also can leverage this opportunity and use new technologies in the purification business to make really cost-effective drugs for their own biosimilar market.
So how does Bio-Rad address these challenges?
So Bio-Rad's process chromatography group is developing state-of-the-art chromatographic materials, and we're really addressing the different needs for different purification steps. For example, we have materials with high binding capacities to be very efficient in the capturing steps of therapeutic proteins. We also have excellent developments in very selective resins, so we exploit the different ligands to bring in interesting selectivities. And well, to give a purification process really the finishing touch, we also rely on resins that are really designed for the polishing step, that rely on the resolution properties of the chromatographic resins. And in this case, we have a couple of good examples there. For example, in the Nuvia S and Q resins, we have integrated the surface extender technology. So in this way, we're capable of increasing the ligand amount and the accessibility of the ligand molecules in the pore volume. And in this case, we excellently increase binding capacity and then also, of course, utilize the excellent hydrophilic chemistry of the Nuvia and Ionosphere resins for that. When you look at unique selectivities that we are having to offer for the bioprocess and biopharmaceuticals, well, there is, of course, mixed mode chromatography. So we've been at Bio-Rad breaking grounds for mixed mode chromatography with the hydroxyapatites, and we have now a lot of different flavors of these ceramic apatite materials there, and the CHTs there. They utilize a cation exchange functionality as well as a metal affinity functionality, and in that way give a unique opportunity for purifications. Other examples for multimodal resins is the Nuvia C'. In this case, we have a combination of hydrophobic interaction chromatography and cation exchange. So this is more like the classical mixed mode interaction, where you will also find the same combination with other vendors. So Nuvia C' is certainly very interesting for certain purifications, because the aromatic group from the Nuvia C' is shielded by the cation exchange group, and in this case we have a less pronounced hydrophobicity on this ligand molecule there, and this makes purification very selective, and also recovery from Nuvia C' steps very good there. Last but not least, of course, we are working with high resolution resins, so we have developed small particle versions of our established Ionosphere and Nuvia beads as well. So good example for that's coming off the market onto the market a couple of years ago is the Nuvia HRS. That's a 15 micron particle version of a Ionosphere based cation exchanger, and this has really excellent properties with respect to the resolution there. It also exploits the bead chemistry of the hydrophilic bead chemistry of the Nuvia and Ionosphere particles to make sure that there is really a very low unspecific binding. So Bio-Rad has been working to exploit three important parameters for biopurification. These are binding capacity, these are selectivity, and high resolution. And with these three parameters, every chromatographer in biopharmaceutical process development can really develop efficient purification processes for his target molecules. This is how Bio-Rad contributes to solving the purification challenges that the modern biopharmaceutical markets have now globally.
Thank you so much, Carsten, for coming and talking with us.
You're welcome. Thank you.